Hi, hello, my dear students. This is your Prasanna Harikrishna sir. So, I am teaching botany. Today we are going to see mechanism of ETS. So, that is electron transport system. In the last class, we have discussed various complexes of electron transport system. Usually, there are 5 complexes: complex 1, complex 2, complex 3, complex 4 and the fifth complex that is called as oxysomes. Okay, so all the 5 complexes are located at the cristae of inner membrane of mitochondria. Okay, so we have discussed 4 complexes and separately we have seen the fifth complex, that is oxysome. Clear? So now we are going to discuss about the mechanism of electron transport system. So what is meant by mechanism? Mechanism tells us about how the electrons move from one complex to another complex and how the protons move from matrix to perimitochondrial space. Clear? So let us get started. So this is about mitochondria. I want to draw the diagram of mitochondria. So this is what the inner membrane, the infoldings of inner membrane are generally called as what? Cristae. Okay, so there are different complexes. These complexes are located on the cristae of inner membrane of mitochondria. Clear? So I want to cut it. I want to cut some part of cristae of inner membrane. Clear? So then the structure will be like this. This is the membrane, cristae membrane, or you can say inner membrane of mitochondria. Clear? So there are different complexes. Usually there are 5 complexes. This is complex 1. So this is complex 1, usually in L-shaped structure. Right? So this is complex 2. This is complex 3. Then come to complex 4. Then finally we have got the last complex, that is fifth complex, or you can say oxysome. This is oxysome or F0 F1 complex. Okay, so these are the complexes. Clear? This one is complex 1. So this is mitochondria, mitochondrial matrix. Okay, this is matrix. The space between 2 layers, this is outer layer and this one is inner layer. Okay, the space between 2 layers is called as perimitochondrial space, PMS. Okay, so perimitochondrial space, this is a matrix. So in which in this particular diagram, so this portion, this entire portion is matrix. This is matrix. Okay, matrix of mitochondria. So on the opposite side to the matrix of mitochondria, we have PMS, means perimitochondrial space. Okay, so this is perimitochondrial space. Okay, then come to complex 1. This is what? Complex 1. This is complex 2. This is complex 3. This is complex 4. So this is complex 5. Okay, so complex 1, complex 2, complex 3, complex 4 and complex 5. So apart from all the 5 complexes, we have got another mobile carriers, that is electron mobile carriers. One is ubiquinone, that is UQ. So this is ubiquinone. Okay, ubiquinone. And one more mobile carrier, that is cytochrome C. This is what? Site C. Site C stands for cytochrome C. Okay. So then this complex 1 is generally called as what? NAD oxidoreductase. So oxidoreductase. I have already told you the entire information regarding to this, all the 5 complexes. Just remember, this is complex 1 or NAD oxidoreductase. This is complex 2, or you can say succinic oxidoreductase. Complex 3 cytochrome BC complex. Complex 4 cytochrome A1 A3 complex, and complex 5 is oxysome. Okay. Then come to number 1. So this has FMN as a cofactor. FMN. So flavin mononucleotides. Here FAD, flavin adenine dinucleotide, is a prosthetic group. FMN is a prosthetic group of complex 1. So FAD is the prosthetic group of complex 2. Then this is what? Site B and site C. Okay. This is site A1. Then this is site A3. Okay. So this is about different complexes. So at complex 1, so NADH2 of Krebs cycle, NADH2 of Krebs cycle, and link reaction. So NADH2 of Krebs cycle and link reaction is participated at complex 1. So it will become into NAD plus. Okay. Then there is liberation of electrons and protons. These electrons move from different complexes. So the movement takes place like this. Okay. So this is, I want to draw the diagram. Just you can have a look. So here FADH2 and NADH2 of glycolysis. So both will participate at complex 2. Then FADH2 will become into FAD plus. NADH2 will become into NAD plus. Clear? Then there is a liberation of electron. This electron will move like this. Okay. Then so these are the electrons. This is what? Electron. This is electron. This is electron. So this is about the complete diagrammatic representation of the electron transportation mechanism in the living organisms. Clear? So now we start from the complex one. See, NADH2, nicotinamide adenine dinucleotides. There are two protons. Protons nothing but it is in reduced form. So what is the form here? Reduced form of NAD is called as NADH2. Okay. So NADH2 of Krebs cycle, NADH2 of link reaction do participate at complex 1, that is NAD oxidoreductase or NAD dehydrogenase. Okay. So then this NAD is converted into NAD plus. This is oxidized form of nicotinamide adenine dinucleotide. Okay. So why it becomes oxidized form? Because it lost 2 protons and 2 electrons from NADH2. Okay. So NADH2 loses 2 protons and 2 electrons. Clear? So then it is converted into NAD plus. Then these electrons move to complex 1. Okay, so complex 1 there is a prosthetic group, that is FMN. So from here these electrons move to ubiquinone. Okay, so from complex 1 these electrons transport to ubiquinone. So whenever the ubiquinone, so this is what UQ plus is called as oxidative form or oxidized form, oxidative stage. Then UQH2 means reductive state. So this is reductive state of ubiquinone. Then UQ minus is semi-ubiquinone or semi-oxidized or semi-reductive form of ubiquinone. Okay. So ubiquinone is one of the mobile carrier. So one of the mobile carrier, ubiquinone is generally a mobile electron carrier. So it is located in between complex 1 and 4, complex 1 and 3 or complex 2 and 3, right? So see carefully the diagrammatic representation. This is complex 1, complex 2 and complex 3. So this is what? Ubiquinone. So ubiquinone is mobile carrier. Mobile carrier nothing but what? It moves from here to here and here to here like this. It moves like this. Okay. So it moves in the 2 membranes, in between 2 membranes of this particular crystal. Clear? So ubiquinone moves from complex 1 to complex 3 and from complex 2 to complex 3. So this is mobile electron transporter or mobile electron carrier. Clear? So now this electron from complex 1 reaches ubiquinone. So whenever the electron reaches this particular ubiquinone in oxidized state will become into reduced form of ubiquinone. Clear? So whenever the electron, for example, this is what? UQ plus. What is this? Oxidized form. So I can use the word O. O indicates what? Oxidized state. So whenever it gets electron, so whenever oxidized form of ubiquinone gets electron from any of the sources, so it will become into reduced form. Okay, this is what? Reduced form. Then whenever it loses, so whenever one electron loses from the reduced state of ubiquinone, it will become UQ minus, indicates semi-ubiquinone. So whenever it gets lost another electron, so then it will become into the same condition or same state, that is ubiquinone plus, means oxidative or oxidized stage of ubiquinone. So this entire cycle takes place in between complex 1 and 3 and complex 2 and 3. So this cycle can be called as quinone cycle. What is this? This is quinone cycle. Okay. So whenever one electron reaches ubiquinone, okay, so one electron or electron reaches from complex 1 to ubiquinone. So 4 protons from the matrix. There are 4 protons from the matrix. Matrix is a reservoir for protons. So these protons move or transport from matrix to perimitochondrial space. Clear? So whenever electron move from complex 1 to ubiquinone. Complex 1 to ubiquinone. 4 protons from matrix transport or move through this particular membrane into the perimitochondrial space. Clear? Now, so this come to complex 2. So at complex 2 you can have FADH2 and NADH2. Clear? So FADH2 nothing but reduced form of flavin adenine dinucleotides. NADH2 we know very well, that is reduced form of nicotinamide adenine dinucleotide. Clear? So FADH2s are formed in the Krebs cycle. I have already told you the entire picture of Krebs cycle in my previous session. Okay, so FADH2 is formed in the Krebs cycle and NADH2 of glycolysis. Clear? So all the NADH2 of Krebs cycle and all the NADH2 which are formed during the link reaction are participated at the complex 1. Okay, but NADH2, two NADH2s are formed in the glycolytic pathway. So glycolytic NADH2 can participate at complex 2 only. They do not participate in the from the complex 1. Remember, clear? So NADH2 is converted into NAD plus, FADH2 is converted into FAD plus. Means what? 2 protons removed from NADH2 and FADH2 and electrons are also removed from these 2 compounds. Clear? These removed electrons move to the prosthetic group of complex 2, that is FAD, flavin adenine dinucleotide. Then electrons move to again, that is ubiquinone. Again this cycle takes place. Okay, so whenever oxidized state gets electrons, 1 electron or 2 electrons, it will become into reduced form of ubiquinone. So if one losses from ubiquinone, reduced form of ubiquinone, so it gets become it gets converted into semi-ubiquinone. Then whenever one more electron is lost from the semi-ubiquinone, it gets converted into oxidized state of ubiquinone. Clear? So now, just have a look, this is electron transportation. So whenever the electron from complex 2 reaches ubiquinone, clear? Whenever the electron from complex 2 reaches ubiquinone, so 4 protons from the mitochondrial matrix move to the perimitochondrial space. So this is another 4 electrons. Okay, whenever electrons move from complex 1 to ubiquinone, or you can say complex 1 to complex 3, there are 4 protons which move from mitochondrial matrix to perimitochondrial space. Clear? Whenever the electrons move from complex 2 to ubiquinone, there are again 4 electrons from the matrix of mitochondria move to perimitochondrial space. Okay, so now these electrons, so this electron from quinone cycle gets moved from ubiquinone to complex 3, that is cytochrome B and cytochrome C complex. Okay, so this is movement of electrons in complex 3. Then this electron move from complex 3 to T3 to cytochrome C. Remember, so this is cytochrome C. Okay, so cytochrome C is the mobile electron carrier, but it is present towards the perimitochondrial space. Clear? So this is also the mobile carrier, but it is located inside the crystal layers. But this cytochrome C is a mobile carrier which is located towards the perimitochondrial space. Okay, now these electrons are received by cytochrome C and they are moved from cytochrome C to the next complex, that is complex 4, that is cytochrome A1 and A3 complex. This is a movement of electrons. Clear? So finally these electrons, whatever the number of electrons, whether it is 1 or 2 or more number, okay, all the electrons reached oxygen and protons finally. So these protons, 2 protons and of oxygen combines together and there is a formation of water. Okay, so what kind of information this electron movement gives us? Remember, the final electron receiver in the entire complexes is complex 4. So complex 4 is a final or terminal electron receiver. Okay, so these electrons are reached to the half oxygen and 2 protons. These the two compounds combine together and there is a formation of water. Clear? So remember, there are 4 protons in the matrix of mitochondria. So out of 4 protons, 2 protons are utilized for the formation of water. Clear? And remaining 2 protons move whenever there is an electron movement in the complex 4. These 2 protons move from matrix to perimitochondrial space. Okay? But remember, here 4 protons moved from matrix to perimitochondrial space. So here also 4 protons already moved from matrix to perimitochondrial space, but here only 2 protons. 4 plus 4, only here 2 protons. 2 protons moved from matrix to perimitochondrial space. Why 2 protons? Why not 4 protons? Remember, already 2 protons are utilized for the formation of water. Then remaining 2 protons move from matrix to perimitochondrial space. Okay. So now just observe this entire picture. This is movement of electron transport system, movement of electrons in the crystal layers. Okay now, so just concentrate more. This is complex 1, complex 2, complex 3, complex 4 and complex 1. So complex 5 does not involve in the electron movement, my dear, remember. So electron movement ends with complex 4 only. Okay, there is no involvement of complex 5, that is oxysome, in the electron movement. Okay, now there are 2 mobile carriers. Out of 1, 2, 3, 4, 5 complex, there are 2 mobile carriers. One is ubiquinone. This is mobile carrier and it is located inside the crystal layer. This is another mobile carrier that is located towards the perimitochondrial space. Clear? So whenever electron moves from one complex to another complex, so 4 protons from matrix, 4 protons of matrix move towards the perimitochondrial space. 4 protons, but here 2 protons. Why 2 protons? 2 protons gets utilized in the formation of water. Clear? So this is about complete movement of electrons in the electron transport system, or you can say crystal. Okay, so this is about today's session. So in next session we will discuss oxidative phosphorylation. What is that? Oxidative phosphorylation. How the ATP is synthesized. Clear? So we will see it in the next session or next class. Thank you very much.